And Rubayat, you can go ahead and share your screen, Rubayat, when you have a chance.
Okay. All right, I hope everyone can see my slides.
Yeah. So we're gonna wait. The webinar participants are trickling in now. It takes a little while for everybody to trickle in.
All right, let me know when I can start.
Okay, I will. Well, we're gonna wait another minute. And
Sure.
good morning, everybody. Welcome to the webinar. We are just gonna start in maybe a minute or so. We're waiting for all the attendees to trickle in to the webinar. Okay, we're going to go ahead and get started. So before Rubayat starts, I want to welcome everyone to the NSF funded Research Collaboration Network webinars. These webinars are for what we're calling junior researchers, including assistant professors, postdocs, and PhD students. And the goal of the webinars is to bring the community together, to share information beyond the anecdotal information that we can share briefly through emails and Slacks before we have publications, and really just to celebrate the hard work of our community during this time where it's very challenging to do research. I say this every time. Sometimes it's challenging to get pipette tips. Sometimes it's challenging to get out of bed. So challenging in very different ways. We hope everyone had a nice, restful Thanksgiving weekend. And as we head into the holidays, we wish you all a happy holidays. This is our last webinar of this calendar year, but we'll be starting up again in early January. For those of you who are attending who have would like to present, are an assistant professor, postdoc, or PhD student, please send a note to me or any of the PIs or Jill McClary, who is a postdoc working on this project. Okay, so with no further ado, we have two speakers today, which we're very grateful for their time to join this webinar. The first speaker will be Rubayat Jamal from University of Utah, and our second speaker will be Yihan Zhang from UC Davis. So we're going to start off with Rubayat. Just a reminder on how this works. The speaker will speak for about 20 minutes. You all can put questions into the Q&A, and you can upvote questions with the little thumbs up sign. And then Jill will handle the questioning for Rubayat. So with no further ado, Rubayat, you can go ahead and begin.
Thank you. So hello and good morning, everyone. My name is Rubayat Jamal. I'm a graduate student at University of Utah, working in Dr. Vedas's lab. So today I'll be talking about our ongoing project on informing COVID-19 disease burden by monitoring the SARS-CoV-2 RNA in wastewater. So I had participated in this project earlier this year, but now we have students and faculties from different universities as well as members from different government organizations working in this project. So my PI, Dr. Jennifer Wittes, she's doing a wonderful job in coordinating the whole project. So by now, we all know that the SARS-CoV-2, which causes the coronavirus disease, is shed in feces, and the viral RNA can be detected from the wastewater. So the results I'll be presenting here is from a 9-week-long wastewater epidemiology study back in April and May from 10 wastewater treatment plants across the state of Utah, serving like 39% of the whole state, and or in other words, like 1.2 million individuals. So in this presentation, I'll be talking about the sample collection, processing, and analysis. And I'll show you some of the preliminary results on solid versus water viral concentration and some inter and intra-laboratory analysis. I'll also give you some results on sewer-shared and sub-sewer-shared sampling, and at the same time, viral RNA decay over time. And finally, I'll give you the outcomes from our state of Utah data and some of the data from University of Utah campus. So this is the representation that helps us to understand how we classify our data based on different sewershed. For example, we are collecting samples from these wastewater treatment plants, influent. But at the same time, we are also collecting samples from different interceptors. Now, we can categorize these sewer sheds based on their size, population, and the activity going on in this different sewer shed. For example, the greens means like all these places, they don't have the virus, and the red means like all these individual households or residential place, they have the virus. So if we just collect the sample from the wastewater treatment plant influent, there is no way to tell how the individual sewer sheds are performing. So that's why to have that in mind, we collected the sample not only from the influent of the wastewater treatment plant, but also from the interceptors, which represents different sewer shed. So this is the basic flow of our sample collection, processing, and analysis. So we receive the 24-hour composite sample that has been transported to our lab through a ice bucketed container from different treatment plants and the interceptors. After receiving the sample, we create some subset of samples and spike them with beta coronavirus and inactivate them at 65 degrees for 1 hour just to make sure if there were any SARS-CoV-2 RNA or SARS-CoV-2 in the sample that is not affecting the lab personnel. So after that, we centrifuge our sample at 4,000 G for like about 20 minutes, and then we transfer our supernatant to a new container, and then lower the pH to 3 to 3.5. Then we use 3 sets of filter paper to filter like around 100 ml of wastewater sample. So the first filter paper, we store it at -80 for future use. And the second and third filter paper, we use it for RNA extraction. So in our lab, we use the manual RNA extraction. It takes around like 4.5 to 5 hours, but it is much better than any of the commercially used RNA extraction kits. After that, we use the triplicate qPCR for detecting the SARS-CoV-2 and the OC43 genome from the sample. And finally, we analyze our results and present them as viral gene copy per capita per day in the sewer shed, and we inform the outcomes to the health department. So I will talk about this viral gene copy per capita per day in my next slide. But at the beginning, we were interested about how the percent of detect in wastewater influenced solid compared to the liquid, and the solid is retained in the centrifugation tube bottom after centrifugation. So here in this figure, the orange bar are the percent of detect in liquid samples, and the blue bars are the percent of detect in the solid samples. So we determined the gene copies per gram of solid and per gram of liquid in 8 samples. So overall, the SARS-CoV-2 was detectable in 91% by mass, in liquid samples and only 9% by mass in the solid phase. So based on these results, we decided to move forward with the liquid sample. So at that time, there were 2 other labs in Utah who were helping us with the sample processing. So we performed inter and intra-laboratory replication analysis of split samples to compare the sample processing in between the labs. So here, samples were collected from different treatment plants, as I mentioned here with facility A, B, and C. And the viral gene copy detected in individual labs were presented in the box, in this box. So we found mean among the labs were 104, 210, and 90 gene copies per ml. We were also tested for the variance and found that the results are not statistically different in between the labs. So that suggests us like the data is comparable between the labs. So as I mentioned before in my previous slide that we have samples coming from 10 different treatment plants. So the time to account, so we have to account for the flow rate and the population from different cities. And then that's why we have expressed our results in million viral gene copies per capita per day. And we use this equation to present that. So this is the map of the sewer-shed and sub-sewer-shed in our study area. So Central Valley that receives the sample from all these sub-sewer-sheds. And in this figure, this is the main line that goes through the city for sewage sample. So all these sub-sewer-shed, they are different based on their size, their population density, and the activity. Some of them has more residential place, some of them has more industrial activities going on. So we have collected the samples from the Central Valley, which is the main treatment plant here. At the same time, we have collected samples from all these different sewer sheds. So let's talk about the equation that I showed like a couple of slides back. So these are the factors that are being used in the equation. You have the average flows, the population in the individual sewer shed, and you divide that with the population and get the per capita flow in the sewer shed and feed all this information in the equation. And we also take in consideration of the inflow infiltration in our results. For example, here, the KRN and SSL, they have almost the same types of flow, but because of the population density, we can see there's a huge change in the average gallon per capita per day. So we suspect that the infiltration inflow are mainly factoring this whole equation. And at the same time, you can see when you take in consideration of the inflow infiltration, the number, the average million viral gene copies per day also increased a lot. And that gives us some false higher results as we can verify with the actual case number in that situation. So the findings from the previous slide is presented here. So plot A represents the viral gene copy where the flow rate has taken into consideration, and plot B is the same results where flow rate has not taken into consideration. So different interceptors show variable gene copies compared to the wastewater treatment plant results, which is here. And the reason could be number one, different disease burden in different sub-sewer-shed sampling, or higher travel time for the interceptor to the wastewater treatment plants causes RNA to decay. So we can say sampling interceptors within a larger area did reveal finer resolution of the disease burden. So now we know like numerous physical, chemical, and biological factors could influence the persistence of viral RNA in the wastewater. So in this study, we evaluated the decay of RNA over time at different temperature. So overall, the results represent that the 4 and 10 degrees Celsius when the samples were stored, we still can detect the viral RNA over 22 to 48, 22 to 24 hours. But when we stored that same sample at 35 degree, we couldn't detect anything after 6 hours. So that means like the SARS-CoV-2 is more liable to temperature and uh, external factors compared to other previously reported coronaviruses. So based on the results I have presented so far, the recommendations are as follows. So the shortest hold time possible prior to the RNA extraction is recommended. We have to account for the flow rates to facilities. And we also have to take consideration of sub-stewardship sampling and consider the inflow and filtration. So some more results from the Utah pilot study. So average and the standard deviation of viral RNA at different temperature are presented here with the bars, and the red line presents the weekly COVID-19 cases. And the vertical dash here that represents the first week of sampling. So it seems like the communities with the higher case number also had a higher viral load in the wastewater. But we were also interested to find out like how that correlates, how the SARS-CoV-2 viral gene copy correlates with the COVID-19 disease burden. So in plot A, the viral gene copy in all the sewersheds were plotted against the daily new COVID-19 cases in each sewershed. And we find out a significant correlation. In plot B and C, similar trends were also observed when the case counts were compared with the weekly COVID-19 disease burden, but not all the sewersheds followed that trend. However, when we take in consideration of the 1 week lag in the COVID-19 cases, we found some sort of correlation. So moving forward, we were also interested to know how the case number is related with the SARS-CoV-2 shedder in a stewardship. So at the time of the study, some of the critical information is like how much virus RNA is being shed at the onset of the disease, how much is shed during the peak hours, and how much is shed during the decline of the disease. So we used available literature data of 4.7 log gene copies per ml of feces, and we have estimated the number of ill individuals and compared that with the confirmed COVID-19 cases. And we found a correlation between these two factors. Currently, University of Utah and 2 other universities located in the northern and the southern part of the state is working with the Department of Environmental Quality to monitor the viral load in 42 facilities throughout the state of Utah, or in other words, like 80% of the Utah state population. So the daily COVID-19 case and the million viral gene copies per capita per day, and some trend analysis were found in the website wastewatervirus.utah.gov. Here are some of the claims of our effort to University of Utah campus sampling for COVID-19 disease. So we have over 35,000 students in the University of Utah, and 13% of them are staying in the college-owned dorms. So we have stated the surveillance process. We have started the surveillance process back in August this year, and it's an ongoing process. So we have looked at several high traffic areas throughout the campus, and we collected the sewage sample from those spots. So the university is some sort of not comfortable with sharing the data, but I will share some of the data here. So there are like these red lines, they're representing the weekly COVID-19 cases from the community, and the black dots, they're representing the viral gene copy we collected in the same time from the same area. So they somewhat represent, like correlate with each other, but this gives us a good idea about the current and future outbreaks in the area. So this work would not be possible without the contribution from the university faculty members, students, local DQ personnel, and the wastewater treatment plants. So I'm not going to mention all the names here, but they are presented here. So thanks everyone. I'll appreciate your time and happy to answer your question.
Thanks, Rubayat. I'm going to go ahead and move us into the Q&A. I'll just start at the top of the list here. So the first question is about your flow data. So do you have an understanding of flow in the different sewer sheds and whether or not it correlates to population or just how variable those flows can be?
Yeah, so we have flow data from different sewer sheds. So we have flow data from the wastewater treatment plants, and at the same time, we also have the flow data from the interceptors. And if you look at the equation, yes, the answer is yes, they do correlate and they do have an effect on the ultimate outcome. Because based on the flow, based on the flow, we can use the population in that sub-sewer shed, and we can get ultimately to our end result of the equation, where we express our result at 1 million viral gene copies per capita per day.
Okay, and I think another follow-up that just popped in kind of related to that, are the sewers in this area combined sewers? So do you have inclusion of stormwater in some of these sewer sheds?
Yeah, yes.
They're all combined, okay.
Yeah.
Thanks. And I had another question come into the chat about the decay parameters. So could you describe a little bit more the test conditions that you used to perform the decay study and how you detected the RNA from those samples?
Yeah, so I believe the question is about this slide. So we collect the samples, and then we transfer like 50 ml individual tubes, and we change the temperature of the samples. So some of them are treated at 4 degrees, some of them are at 10 degrees, some of them are 35 degrees. And we have, from those samples, we have collected more samples over time, and we tested the qPCR. So over the hours, we were estimating how much we can detect from individual hours, and then we plotted them against our and the gene copies, viral gene copies, and that's how we came with this result.
And do you know how the decay rates that you've found so far compare to any other previous studies? I think there's been a couple.
Top of my head, no. At the time we were doing this, I don't think we have seen any other data, but maybe there are a few more data now. But we can say that like it is much more temperature sensitive that we have seen previously with other coronaviruses. And we were a little bit concerned about it because it takes, in a small city, it takes around 1 to 3 hours for the wastewater to reach the treatment plant. And from there, people can just do the sampling. So in that time, the average temperature could be, like, for example, in our city, average temperatures are around 11 to 15 degrees. So we were concerned about how much viral RNA we are losing because of this higher temperature. And that was the motivation for us to do this study. And we have seen that, yeah, if you increase the temperature, there will be a significant loss.
Okay. Another kind of technical question. People are wondering what your general OC43 recovery rate is for your method.
Yeah, so we did the recovery for OC43 and murine hepatitis virus, and it was similar with what we have found in the other literature reported value, like 26 to 30%, or I would say like 25 to 30% based on different study conditions.
Okay. And let's see. There's one more related to your different sewer sheds. So, and I have a follow-up on this one too. Do you have any information about the different characteristics of your sewer sheds on, so for example, socioeconomic indicators? So do you expect those factors to differ between your sewer sheds?
Yeah, so the data we collected from a government website, and they provide us with the population, like the percent of urban and the industrial activities and all those related information in this website. And we also factor in the fact that infiltration can also cause the flows in this subsewer. But yeah, we have some preliminary results and data about the factoring of the subsewer.
Okay, yeah, and sort of a sort of follow up on that, since you've been providing some of this data, I know to the health department, you said, curious if there's been, has the health department shown interest in kind of managing the response in different sewer sheds? Do they find value in seeing those different areas of the city that you're able to identify with your sewer sheds, or are they kind of, I've heard of different instances with those boundaries being somewhat arbitrary from the perspective of public health, but have you seen those kind of boundaries to be useful for their interpretation?
I'm not exactly sure what kind of actions they're taking, but they're interested very much in this data, and they're meeting Dr. Vedas twice a week and trying to understand like what kind of forecast, what kind of precaution they can take based on the data I have. So my answer is yes, they're interested and they're taking actions in these subsewer areas, but I am not 100% sure what specific actions they're taking. So it could be anything from like public health measures like lockdown, or strengthen the restrictions from all of these up until the point where they're factoring the case, like they have to manage their sewage lines to reduce the infiltration. But there could be a lot of things going on.
Great. I think we have maybe just one more question here, unless any other ones come in, or a couple. So the first one I wanted to ask was about your comparison between confirmed cases and estimated shedders. Can you go over again how you determined the number of estimated shedders?
Yeah, so we have, I think at the time we were doing the study, there wasn't much study about like how much virus is being shed in the onset of the disease, during the disease, or after the disease has declined, how much virus is shed in individual events. So we found one literature where they were presenting like 4.7 log gene copies per ml of feces. And we have used that data to transfer that to estimated the individual shader in that area. And that we correlate with the confirmed cases. So it comes from the individual shader, from the amount of virus being shed in individual event, transfer that to the individual shader, and correlate that with the confirmed cases.
And then maybe we have time for one final question that came in here. And I had this related question too about your data between the solids and liquid phase. So other researchers finding a larger partitioning of the virus to solids. Do you have any thoughts on why you observed lower number of positive samples when you were analyzing the solids versus the liquid phase?
Yeah, it could be. So just to clarify, so we received the liquid sample and we take like 300 ml or 400 ml of sample from the actual bottle, and we centrifuge that over time at 4000 G for 20 minutes. So this is not like a solid sample directly from the wastewater treatment plant. This is the sample that is being mixed with the liquid. So based on the turbidity of the sample, the percent of solid can be different. So it could be different from individual day, from individual treatment plans. So at the time we were interested about, we were thinking about, hey, we are taking only the liquid sample, but are we missing something in the solid after the centrifugation? So that's how, that was the motivation of doing this study. And we found these numbers, but it could be different. It could be different based on different treatment plans where they have more chunky stuff in their sample. It could be different because if you centrifuge the sample even harder. So a lot of factors can be involving here, but this is what we got with our sample in Utah.
Okay. Yeah, and I imagine maybe also a factor of limits of detection playing in there then too, since it's not a specific solid sample. So,
yeah, could be.
Thank you very much, Rubayat. That was very helpful.
Yeah, my pleasure.
And I think we'll pass it over to our next presenter, Yihan. Go ahead whenever you're ready.
Good morning, everyone. My name is Yihan, and I'm a PhD student from the Department of Civil and Environmental Engineering at UC Davis. So today I want to present quantitative microbial risk assessment of SARS-CoV-2 in wastewaters. So this is a joint work between UC Davis and UC Merced, and here's a picture of our team. So today I want to break down my presentation into 4 parts. First, what is the motivation of our project? Second, what is a quantitative microbial risk assessment? And what is currently missing in literatures? And lastly, I want to present our methods and the preliminary results sessions. So the motivation of this project is to protect wastewater treatment plant workers' health. As we all know now that COVID-19 is a respiratory disease that has caused a global pandemic. And currently to date, there's no evidence showing that anyone has become sick with COVID-19 when dealing with wastewater treatment. But wastewater treatment plant workers do have the potential of being exposed to wastewater pathogens, including SARS-CoV-2. So SARS-CoV-2 from the infected patient can enter a wastewater treatment plant through the household sewage systems. And during the early stages of the wastewater treatment process, for instance, the preliminary treatment process and the primary treatment process, the concentration of virus in wastewater are pretty high. So SARS-CoV-2 is and the pathogens can be aerosolized too. And SARS-CoV-2 is one of these respiratory disease, and its exposures to wastewater treatment plant worker can depend on many different factors. For instance, their inhalation rates, then their duration of the exposures, and the concentration of the virus in the airs, as well as the dose response functions. Since now we know what the risk factors are, we need to think about what the response could be when being exposed to SARS-CoV-2. So the response of being exposed to SARS-CoV-2 can be infections, but also depending on the susceptibility of wastewater treatment plant worker, the response can end up being illness or even death. So how do we protect wastewater treatment plant personnel from exposures? We want to conduct quantitative microbial risk assessment to estimate the risk or the probability of the health effect. Here, I want to emphasize that to date, no evidence shows that SARS-CoV-2 can be transmitted through wastewater, but we do want to use SARS-CoV-2 as example to illustrate the potential exposure to wastewater pathogens, so we can be better prepared for future waterborne outbreaks. So a standard QMRA usually includes 4 steps. The first step is hazard identification, in which we identify the type of the pathogens, as well as the concentration of the pathogens in wastewater. And the second step is the development of a dose response curve. Usually we use the data from the epidemiologic studies, and I will use an example to illustrate this process later. And the third step of the QMRA is exposure assessment, which touches on the areas I mentioned previously, such as exposure pathways, duration of exposures, and the inhalation rate, and so forth. And then lastly, we want to combine all this information together to do a risk characterization, risk management, so we better characterize the risk and better manage the risk so that we can protect wastewater treatment plant workers' health. So now we want to take a closer look at each step of the QMRA. As I mentioned previously, the first step is hazard identifications. So in our case, it's going to be pathogen identifications. In hazard identifications, we want to work with published data about wastewater-based epidemiologies, so we can get more information about pathogen sheddings, as well as the pathogen concentrations in the influent and how it changes throughout the wastewater treatment processes and how it varies depending on the seasons. The second step of the QMRA is the development of the dose response curve. So a dose response curve describes the relationship between the exposure dose and the probability of the response. And this data is usually derived from the epidemiologic studies. For instance, researchers challenge a group of animals with different concentration of the virus, and they observe and record their response. I will use an example to illustrate how researchers develop the dose response curve from the epidemiologic studies. So there's a virus in the wastewater, and if we know how much exposure dose a treatment worker is being exposed to, we can plug in this exposure dose into the dose response curve to predict the probability of the health effect. So there are two common types of the dose response models. The first one is the exponential models, and the second one is the beta Poisson models. So the difference between these two models is that exponential model assumes that organisms have identical and independent survival probabilities, but the beta Poisson model assumes that this survival probabilities is changing between different organisms, and it follows beta distributions. So next question you might ask would be, how do we fit the experimental data from the epidemiologic studies into each of the dose response models and determine which model better describes the data? So to answer these questions, we have two steps. So first, to fit the data, how do we fit the data into each of the models? We want to find the best fit parameters so that we can minimize the deviance between the observed experimental data and the simulated data. The second, how do we compare which model better describes the data? To answer these questions, we want to calculate and compare the goodness of fit values between two different models so that we can decide which model better fits the data. Now, I want to use an example to illustrate this process. A study in 2006 wanted to evaluate the susceptibility of mice to the virus MHV1. So to do that, they divided the animal into 4 different groups, and each group contained 5 different animals, and they challenged each mice group with different concentration of the virus. The concentration varied from 5 PFU all the way to the 5,000 PFU, and the researchers record and observe the response. So to use these data to develop the dose response curve, as I mentioned previously, we have 2 steps. First, we want to fit the data into each of the models independently by minimizing the deviance. By doing that, we are able to find the best fit parameters for each of the models. For instance, the best fit parameter for the exponential model equals to 469, and we can calculate the best fit parameters for the beta Poisson models. The second, how do we determine which model better describes the data? We want to calculate the goodness of fit values of each of the models. For instance, the exponential models, the goodness of fit values equals to 0.89, but for the beta Poisson models, the goodness fit values equals to 0.76. Because 0.89 is greater than 0.76, we can draw the conclusion that for these data sets, we will use exponential models to fit the data. The exponential model better describes the data. The third step of the QMRA is the exposure assessment. So the objective of exposure assessment is to estimate the magnitude and the frequency of exposure to pathogens through the identified exposure pathways. Usually there are two types of the exposure assessment. One is the public exposures. For instance, when we eat in a restaurant, we have the chance being exposed to a virus. The second one is occupational exposures. And today we will only be focusing on occupational exposures. And Within occupational exposures, there are two things that will impact wastewater treatment plant workers' health. One is the infrastructures, where they work at, and the other one is the occupational safety factors in the facilities. Within the infrastructures, there are working units and environmental conditions, and what equipment they used will impact wastewater treatment plant workers' health. Within the working unit, for instance, if the treatment plant workers is working in the collection systems or they're working in the pre-treatment and primary treatment process, they have a higher risk of being exposed to virus. Within the safety factors, many factors would also impact wastewater treatment plant workers' health. One is the frequency of exposures, and the other one is the use of PPEs, as well as other engineered safety controls. The fourth step of QMRA is risk characterizations, in which we calculate the risk of the health effect. So in the fourth step, we will combine the exposure assessment from step 3 with the dose response curve we developed in step 2 to determine the risk of the health effect. So the health effect can be infections, illness, or even death. So the dose is derived from in these equations, the dose is derived from the exposure assessment, and the dose response parameter is derived from the dose response curve that we develop. So far we have covered the basic structures of QMRAs, and moving to question number 3, what is currently missing in literatures? So to date, there's no dose response curve available for SARS-CoV-2. So during the current pandemic, many studies have relied on the dose response curve developed for SARS-CoV-1 to estimate the exposure risk of SARS-CoV-2. This can lead to problems because studies have shown that SARS-CoV-2 is more transmissive than SARS-CoV-1. So if we relied on the old dose response curve to estimate the risk of SARS-CoV-2, we may very likely underestimate the exposure risk. And the second limitation of the old dose response curve is that the end point of the old dose response curve was illness. But ideally, we want to develop 3 different dose response curves. One is for infections, and one is for illness, and one is for death. So for our method sessions, we want to develop a new dose response curves using the recent epidemiologic data, and we also wanted to conduct a new exposure assessment. For the dose response part of our studies, we are currently re-evaluating the recent publications for SARS-CoV-2. So we want to develop a dose response curve for each endpoint, including infections, illness, and death. And we will also validate this dose response curve using the outbreak data. So for the exposure assessment part of our project, we have sent out a national survey to wastewater treatment plant personnel. And in the survey, we ask questions about their duration of work, their working locations, and their use of PPE, and so forth. We want to use this data to evaluate their occupational exposures to wastewater pathogens, and eventually we want to combine these 2 sets of data together. So we want to combine the dose response curve with the exposure assessment together to estimate or to predict the risk of wastewater treatment plant personnel during this pandemic. So moving to our result sessions, currently we have developed a dose response curve for SARS-CoV-2 using recent epidemiologic studies. In these studies, the researchers challenged serum hamsters with SARS-CoV-2. So they divided the serum hamster into 5 different groups, and they challenged each group with different concentration of SARS-CoV-2 and observe and record their response. So the endpoint of this dose response curve is illness. So we fit this data into the exponential models by minimizing the deviance between the observed data and the simulated data. And the calculated k-value is only 8.5, which is smaller than the k-value of SARS-CoV-1, indicating that SARS-CoV-2 is more contagious than SARS-CoV-1. And the goodness of fit values for this dose response curve is 0.96, which is greater than the critical value of 0.05, indicating this model well fits the data. So since now we have the dose response curves, we want to integrate it into the exposure assessment. So we sent out the survey to wastewater treatment plant personnel to see what is going on in wastewater treatment in wastewater treatment plants. So in the survey, we ask about what is your primary working locations, and 50% of them indicated in blue answered that they're performing routine plant operations, including manual valve operations, wastewater collections and samplings, and 37.5% of them answered they're working in the collection systems. So this means that a lot of wastewater treatment plant workers have the direct contact with wastewater. So it also means they have the chance of being exposed to wastewater pathogens. And the wearing PPE will reduce this kind of exposure risk. So in the next part of our survey, we ask about what kind of PPEs do you wear when you performing your daily duties. And only 25% of the workers answer they are wearing gloves, and only 23% of the workers are wearing safety glasses and goggles. So currently, we only got about 100 response from the different wastewater treatment plants, and we are still waiting for more people to participate in our surveys. So once we have the survey and the QMRA data, we will integrate our results into the spatial geospatial analysis to evaluate the risk of exposures in disadvantaged communities living nearby the wastewater treatment plants. So the UC Davis team is mainly responsible for the survey and the QMRA part, and the UC Merced team will be responsible for the geospatial analysis. And this is what we have so far. So we are still waiting for more data to come out, and we are still waiting for more people to participate in our surveys. But I want to thank CICHIS for providing us a funding source, and I also want to thank the CWEA, WEF, and CASA for helping us distributing the surveys. Here's my contact information, and now I'm ready to take questions.
Great. Yeah, thanks so much. It's really informative. Fascinating how you try to apply and work with the limited information that is still available, unfortunately. Let's see what we got here. So trickling in some question. In your analysis, what data are you using to characterize the concentration of SARS-CoV-2 virus in raw wastewater?
So if I ended, oh, okay. So we use the published data from the literatures.
Okay.
To get these informations.
Okay, good. I have a question from Ellie coming in. Ellie, unmute yourself.
Yeah, sorry, it's weird. The panelists can't enter questions in the Q&A, but I was wondering, the wastewater treatment plant worker exposure work that you're doing and the surveys you sent people. So I have a couple questions. The first is, did you have to get IRB approval for that? And the second question is, how are you going to analyze and present that work so that it can be useful in the future? Because I feel like that exposure information could be useful for a lot of people interested in occupational health applications for wastewater treatment plant workers beyond SARS-CoV-2. So that's kind of a long question. I hope you remember it all.
All right, good. So a two-part question. First of all, so did you file an IRB when you approached the wastewater workers for their behavior?
Yes, we do file the IRB before we conduct the surveys.
Good. And the second one is how applicable, how do you see this work being implemented, Yihan, in the future? And not necessarily only for SARS-CoV-2, but also for other diseases possibly.
Yes, so as I mentioned in my presentations, now we know like SARS-CoV-2 there's no evidence showing SARS-CoV-2 is transmitted through wastewaters, but we do want to use this as an example so we can be prepared for other wastewater pathogens. So in the survey, we ask many different questions like what is your primary working locations and the use of PPEs, etc. And we hope that we can identify like different concentrations, and we want to know like if this exposure concentration is associated with their working locations and etc. If this answers the questions.
Yeah, thank you. Well, let's revisit Jeff's question. He said he still is uncertain of what specific data are going into the model with respect to the concentration of SARS-CoV-2 in raw wastewater. And I'm not sure what the question is. Is it the method, how you measured the virus? Of course, there's many methods. Jeff, if you want to, if you can further.
Yeah, I'm gonna allow Jeff to talk here so I can.
Awesome.
Okay, Jeff, go ahead.
Can you hear me?
Yep.
Oh, cool. Yeah, so I'm curious as to specifically what data, not viral RNA, but what data are you using to characterize the concentration of live virus that could infect workers in your analysis? I mean, you answered that it came from the literature, but I'm curious as to specifically what data are you using and how are you characterizing the concentration of virus in wastewater? How do you do that?
So our study has two parts. One is a QMRA part, and the QMRA part, we will use the epidemiologic studies to develop the curve, to develop those response curves. But for our studies, for our project, we're not measuring the concentration of the virus in wastewater, and instead we're taking the concentration from literatures to evaluate the risk of exposures for wastewater treatment plant workers. So for us, we are not measuring the concentrations.
Okay, if we haven't fully resolved that, Jeff, I encourage you to reach out to Yihan. There's a few other questions that I think that come up. Yihan, I think we all like models. Models are fast, they can be informative, they are not dangerous, you don't have to expose yourself. But also people say garbage in, garbage out, and all models are wrong, some models are useful. When we read these studies, we never quite know where the key assumptions are that one must question. So, but now that we have the author here, can you tell us, as you look at your work, you have a selection of a model, you have certain data inputs, and some of them you create, others you take from the literature. Where do you feel is the greatest uncertainty injected that might call into question the number that gets spit out by the model? So again, if you look at the model, what drives uncertainty? If there's one thing that you could really improve in your model, and whether it's a part of the model or whether it's part of the data inputs, the parameters you put in, where do you feel is the biggest, change or uncertainty injected in your analysis?
I think the biggest uncertainty will be the choice of the models, because like I said, exponential model and the beta Poisson model are the two common types of models, but the uncertainty is that which model better describes the data. So I think the choice of the models is the biggest uncertainties in my studies.
Okay. And maybe let's stay on this slide here. So you have a curve here, but 3 points are essentially just on one. So 2 to 3 data points are driving the curve, and you have a good fit value. Now, as we know, the less data you have, the better the fit, right? In extreme, you get 2 data points. So this is a point where, how big is the uncertainty here? Do we know how much information we have?
Yes, as you mentioned, another uncertainty of this kind of data is that we have very limited information about the dose and the response, because currently people are focusing more about like the susceptibility of the animals, but they're not too much interested about the dose response part. So the current study is just to tend to challenge the animals with high concentration of the virus, so they can like observe what the response could be. So I think another challenge for our studies is that we have very limited information about this dose response data.
Yeah.
And
I also agree with Ali that we probably can learn much from these investigations that goes beyond just SARS-CoV-2. And I'm wondering, have you kind of scrutinized the literature to learn how much of an elevated risk wastewater workers really suffer from, drug-resistant infections, various respiratory viruses, and so forth? What do we know about, how dangerous this job really is compared to working in a meat packaging plant or the assembly line somewhere else?
So you're asking like, what is the risk of working
Well,
in
there's an
wastewater treatment?
yeah, look, there's an underlying assumption that if you work at a wastewater treatment plant with so many potential hazards coming by all the time, that you have an elevated risk. I'm wondering, what is the data foundation for that assumption? Are there really, is there a known increased risk by a factor of 2, 4, 10 for any given, respiratory disease, drug, resistant infection? I mean, all the things that we typically get sick of, do the wastewater worker on average, has a much higher risk as documented by numbers?
I would say I might expect wastewater treatment plants workers to have a higher exposure risk because there are so many pathogens in wastewaters. So, but I don't have evidence to like prove that.
And I think that is the issue. So because we have an underlying assumption that they are exposed potentially to so many bad things because just about anything is in sewage. But in reality, does this really translate into someone who is more often sick, who dies prematurely, who develops diseases more frequently and more severely? I don't know what the literature is. I'm sure there's probably a body of literature for various diseases, but I think it would be important for a study like yours to have that kind of foundational data behind you, because that adjusts the expectation a little bit as you do this, because you couldn't find anything before. Don't be frustrated if you can't see anything in this disease, because it just mimics maybe what you have with a lot of other afflictions that we can suffer. I have one more question. How many people responded to your survey?
So far, we have about 110, and we are looking for about 500 people to participate in the surveys.
That's great. That's great.
Yeah.
Well, I can't wait to see those data in the literature. I think a lot of people would benefit, and I think we're out of time, Yihan. So I'd like to thank you, and I hand it back to Ali.
Yeah, thank you so much.
Yeah, thank you, Yihan. That was great. And I think the reason you have so many questions is it's quite a controversial topic and has, can be very important. So that's exciting for a PhD project for sure. Okay, so we're done. Great to see everyone. And thanks again to Rubayat and Yihan and for all the participants. Have a great rest of your week and see you in January. Goodbye.
Bye.